Alright, what we're going to talk about are contact plates and how bacteria are ubiquitous. So here's a contact plate. This plate has a grid to help you count colonies if you're looking at quantifying it. Contact plates are special because they have a very small lip on the side. When you pour the agar in, the agar upraises on the top of this so that all you have to do with the agar is take this, place it down, lift it up, and you get bacteria. We have here contact plates where we incubated them for 48 hours from a public toilet, the palm of your hand, a handrail on stairs, a can, computer mouse, yoga mat, elevator button, chip bag, and makeup sponge. Now we're going to look at each one of these here. I'm going to pull this over. We can look at our makeup sponge. All of these are bacteria on here. Each circle then is a colony. The colony was formed by a single bacterium which then duplicated into the whole colony. The original bacteria were called colony forming units, or CFUs. Here's a nice spreading colony here, and then here are your normal circular colonies. Let's look at our chip bag here. Okay, very interesting. We can definitely see different morphology of those colonies. There is an elevator button. Okay. Interesting, a lot of spreading colonies on that one. A yoga mat. Our computer mouse. This one's interesting. There on the right, all of that white powdery stuff is a fungus. So big old fungus. You can see all those spores on top of that fungus there. Okay, the other items in there are bacteria. Different types of colonies on that one. Here is a can. A lot of different colonies on that one. Kind of cool ones. The handrail of stairs. Okay, really cool. The palm of somebody's hand. Tons of bacteria on that. And finally, a public toilet. And we don't see any bacteria hardly at all because public toilets get disinfected all the time. Okay, now we want to look at the cheek cells. So here's a view under the 4x magnification of the slide that we just made with the cheek cells. These cells are stained with methylene blue. We can see little itty bitty teeny tiny cells in there, and each of them have a little black dot. That little black dot is the nucleus. I'm going to shoot up under here the 10x. We can see them a little bit better. Okay. Then I'm going to go up here under the 25x. It's a little bit better. Finally, then we'll move up to the 40x. Increase the light to increase the resolution and focus so we can see these cheek cells under the 40x objective. Actually, we can see them really clearly here. So there's a nice cheek cell here. The little blue dots are actually bacteria on the cheek cell. We're going to look at those in more detail if I can move this gently. And we can see that cell right there. Okay. Now what I want to do is if I'm going to go up under oil to look at this, then I need to get that exactly in the center. So there's our cheek cell. It's under our 400x magnification. We're now ready to move to the next step, which is going under oil. Okay, so here's our cheek cell under the 40x objective. I'm going to take it under oil, so I put it halfway between the 40 and the 100x objective. I'm adding a drop of oil to the slide. And then I'm going to wheel in the 100x objective into that oil. Very, very, very gently focus. Okay. focus. There we go. I have to slow down on that focus. Look at it. Beautiful. There we go. Nice. So the bigger blue dots then are the nuclei. So we can see 1, 2, 3 nuclei in this image. So there's 3 cheek cells kind of on top of each other. Then you can see all those nice little blue dots. Those are the bacteria in the sample. So a lot of bacteria on these cheek cells. And you can see the, oh man, my bad. So we can see all these tissues. So I gouged my tissue really well. And then we can get those bacteria. So bacteria have a variety of different shapes. You can see a lot of different shapes in here. You can see some are, most of them look like very small rod shapes. A lot of them are in pairs. So you can see how they're in pairs of 2. We have a chain in the upper middle, kind of a string of 1, 2, 3, 4, 5, 6, 7 of them there. We have a couple ones that are a little rounder, which are called cocci, which are rather than rods. Most of these are small rods, but there are a few that are a little bit coccus shaped. So a variety of different ones. You can see how big the nucleus is, right? And then a cheek cell is a lot bigger than the nucleus. So bacteria, you can see how tiny they are compared to the eukaryotic cheek cells in our mouths. This slide is a smear preparation from the bottom of a shoe. It is taken from a single colony on the contact plate, and then a smear preparation was made. We heat fixed it, and then we added methylene blue, and this is the resulting slide. Currently this is under 4x. I'm going to take it, shift in the 10x objective, refocus. I will then shift in the 40x objective and refocus. And finally, we are ready to go under oil immersion. So I need to make sure that I get the oil. I'm going to shift it halfway between the 40x objective and the 100x objective and put a drop of oil directly on the center of my slide. I'm going to shift in my 100x so I don't get any oil on my 40x, and then we will focus on these bacteria. Okay, so here we can see nice blue bacteria stained from the methylene blue. These bacteria are all pretty much the same. So basically we have long chains of rod-shaped bacteria. There we can clearly see 2 individual in a chain. These are all the same species. They come from a single colony, and they're all exactly pretty much identical. Long rods on there. So big long rods that occur in chains.